This is the Panopto for your second set of selective media, as well as your first introduction to differential only media. So a brief recap, the selective media that we have talked about thus far all contain a selective property. So you're either selecting for something that you want to grow, you want to know if it's there, or you're selecting against things that you don't want to grow. So if you were selecting for MRSA, methicillin-resistant Staph aureus, you would put the antibiotic methicillin, or nowadays oxacillin, into the media, and only methicillin-resistant Staph aureus would grow. Those that are not resistant to that antibiotic would not grow, so you're selecting for resistant cells. So this media contains a selective property, and then it also contains a differential property. So that's going to tell you about the metabolic properties of each organism as they grow on the plate. So you might see if they're able to ferment certain substrates or reduce others based on how they look after incubation. And then you also have differential media. So you didn't have any of that last class. Today you will have the only plate you're going to inoculate for differential media. This contains a differential property, but not a selective property. So anything can grow as long as it can use the nutrients present. We're not selecting against anything. So selective media we'll be using, again, we on Monday used mannitol salt agar, McConkey, and hectoin. On Wednesday, so this next class, what this Panopto is for, you're going to be using EMB agar, which stands for eosin Y, and methylene blue. And you're going to be using blood agar, which is differential only. So EMB is selective and differential. Blood agar is only differential. We won't be using chocolate agar because we don't have that. So we will have demo plates for you that show you, in theory, how the chocolate agar should work. We are still going to talk about chocolate agar, so you still have to know this stuff for your exam and your quiz. So don't tune out when we get to chocolate agar. That's still important. Both of these media can be made selective through the addition of antibiotics. So chocolate and blood is what we're talking about. You can add antibiotics to either of these and make them selective. That's what I said on the last slide with the MRSA example. So eosin Y, methylene blue, if you look over at the components in it, you see that peptone has been degraded, there's lactose, and then there are very specific ingredients. However, we don't have the exact ratios of certain components in the gelatin from peptone, so we would not call this defined media. This is undefined because we don't have exact amounts of each component. So gelatin is just the cartilage from cattle, horses, pigs. It's what makes Jell-O solid. So if you thought you were vegan and you're eating Jell-O, that's not the case, unless it's vegan Jell-O. So gelatin is basically cartilage. That will vary based on the diet of the animal, the specific animal, etc. So we do not know exactly what components are there. So the other components of EMB, the selective property, are two dyes. The first dye is eosin Y. The second dye is methylene blue. These both inhibit gram-positive bacteria. So gram-positive cells, the ones that have peptidoglycan on the outside, will not grow on EMB plates. Some gram-positive bacteria, such as fecal streptococci and staphylococci, might grow. It just depends on how this was made. But for your exam and just knowing basic stuff, EMB will select against gram-positive bacteria. The differential property is lactose fermentation. So the thing that will be fermented, the thing that tells you the differences in metabolism, the substrate, is lactose. So we are looking for lactose fermentation. They will utilize the substrate lactose. And again, remember, three things can be produced when we have fermentation. The one we're looking for is an acid that's going to lower the pH, which will change the color of the dye in the agar. So organisms that do not ferment lactose often increase the pH by metabolizing proteins. It's going to be much less noticeable of a color change. So we'll talk about those results in a minute. The indicators are the same as your selective property on this plate. That's not always true. So the indicators here are eosin Y and methylene blue. They are also your selective properties. The color that they form will depend on the acidity of the environment. This media is buffered so that only rapid lactose fermenters, so those that get in there and they're like, heck yeah, I'm going to use this lactose now, not as a last resort, they will produce sufficient acid to create a really green sheen. If you remember, maybe I'm too old, but perhaps you saw the book Rainbow Fish, how it was kind of iridescent. Vigorous lactose fermenters will look iridescent like that, and E. coli is one of the ones that will do that. So for your unknowns, if you have E. coli, this plate is probably going to tell you if you inoculate it correctly. So that E. coli would be up here at the top. This picture is not great. Of course, you have to rotate it to see all the colors, so it just kind of looks shiny here, but it'll look iridescent in person. So your possible results, you can have growth, which would mean you have a gram-negative species, because typically we are inhibiting the growth of gram-positives. If it doesn't change color, then it is not a lactose fermenter. So that's this first one right here. Now this one at the bottom of the plate, you can have growth with a color change to kind of a purpley pink. It's arbitrary, whatever you think you're seeing. It's a color change. These would be gram-negative bacteria because they grew and they fermented lactose a little bit. So that color change happened because the pH changed, but it wasn't vigorous fermentation. You can have no growth, which would be not a gram-negative enteric species. It's going to be gram-positive or something that is not enteric. And then again, your green sheen is your vigorous lactose fermenter like E. coli. Typically that's just E. coli. And then we have blood agar. So blood agar does not have a selective property unless we add antibiotic. We did not do that. So this is just blood agar. Most pathogens can grow on blood agar, but we have some fastidious pathogens, meaning they require certain nutrients that cannot grow on blood agar. We look for those with chocolate, but we're talking about blood right now. So you can think of blood agar or your red blood cells as little buffets. Inside of your red blood cells are just tons of different nutrients that bacteria would love to have. Really good pathogens are able to lyse open your red blood cells and get those nutrients out of your red blood cells. So they're able to break into the buffet and get all the food they want. But not all bacteria can do that. So the ones that can't do that, the nutrients have to either be available outside or they won't grow. Those are the ones that will grow on chocolate agar. Chocolate agar is lysed red blood cells. Blood agar is your solid blood cells. They have not been broken open. So we are looking with blood agar to identify bacteria that can break open the red blood cells, and that will tell us some about their pathogenicity. So again, you have a pancreatic digestive casein. That means this is undefined. We don't know the exact compositions of each element. You also, for your differential media, you're going to need to know the metabolic property being tested. This is on the next slide. The substrate, so what are we looking for to change? What can the bacteria change? And then the indicator, what thing tells us that there was a change. So for EMB, lactose was the substrate and the indicator was the dyes. Now in blood agar, the substrate and the indicator are the same thing. So we are looking for bacteria that can produce the exotoxin, so exo meaning outside, it's secreted, toxin meaning it's going to degrade other cells. The exotoxin we're looking for is a hemolysin, which can break open blood cells. So hemo refers to blood, lysis, breaking open. So we want to know, do these bacteria produce an exotoxin that can perform hemolysis, lysis of red blood cells? So the metabolic property being tested would be blood hemolysis via hemolysins. The substrate here, the thing that's going to change, is the blood itself. Is it able to break open these blood cells or not? So the indicator is also the red blood cells, and we can see them change. It's similar to when you have a bruise. So the initial stage of bruising, that initial leakage of blood from your blood vessels, might look more red. It just depends on where the bruise is, how much bleeding there was, etc. But it's going to look closer to red. And then over time, your bruise is going to start to turn green as your body recycles those nutrients, breaks those blood cells down. You see the green, which is a component of the red blood cell. So we are looking for different types of hemolysis. So this will be indicated again by the red blood cells. So we might have complete destruction of the red blood cells. We might have just some degradation of them or nothing. Maybe they don't do anything with the red blood cells. So blood agar will allow for the differentiation of gram-positive cocci based on their hemolytic abilities. So there are different types of streptococcal species that do different types of hemolysis. They're capable of different types of hemolysis. Same with staph. Staph aureus can be differentiated from other staph with blood agar. And not all staph aureus is hemolytic. So that's another thing to remember. So if you think you have staph aureus, your hemolysis is not necessarily going to confirm or deny that when you get your results from your blood agar plate. So the three types of hemolysis, you have alpha hemolysis. That's that partial destruction of red blood cells. That's the greenish color, so the middle of this top plate. So think of an old bruise. Your red blood cells are still there, but they've been broken down a little bit. Then you have beta hemolysis, that's this first one. That is complete destruction of the red blood cells, so it's going to appear clear. Now, not totally clear like a window, but clear like the agar you've seen thus far. So it's still going to have agar and nutrients. Or you can have gamma hemolysis, this last one over here. That is no hemolysis whatsoever. So your blood on the plate, the red blood cells are still going to be intact. You're not going to see any difference in color of the media surrounding the bacteria as they grow on your plate. Then we have chocolate agar. Now you guys aren't going to be doing this, but it's important to know. So chocolate agar is basically your blood agar, but the blood cells have already been broken open. So all of those nutrients that are sequestered inside of a red blood cell, they are now available. So fastidious organisms, and I believe that's on the next slide, fastidious organisms that require very specific nutrients will only grow on your blood agar as far as pathogens go. They won't grow on anything else. So if you took a patient sample and you put it on just LB or even just blood and nothing grew, that doesn't mean that the sample you took was sterile, your patient doesn't have an infection. That could just mean the conditions were not right for that bacterial species to grow. So a common pathogen in ear infections, Moraxella catarrhalis, it will not just grow on anything. It has to have specific nutrients. Another one you might have heard of was Neisseria gonorrhea. So perhaps gonorrhea sounds familiar. That's not just going to grow on blood agar or LB. We have to put it on chocolate where all the nutrients are available. It's picky, which we call fastidious. We also use LB plates as a control for this. So if you sample your patient, you streak your sample on LB and you streak it on chocolate, if it grows on both plates, so LB and chocolate, it is not fastidious because it did grow on LB. But if it only grew on chocolate and not LB, that's when you have your fastidious, your picky organism, because it only could grow on chocolate. Chocolate's the only thing that had all the nutrients it needed. The red blood cells were already broken open. Chocolate agar is not actually chocolate. They call it that because it looks like chocolate. It is just lysed blood. So what you do is you get a sample of blood, mix it with some agar, and heat it up. And that heating causes the red blood cells to lyse open, looking brown. So think of if you get a cut and you get blood on some like white fabric, the next day or later that day, it's going to look brown, and that's because your blood cells broke open. So Neisseria gonorrhea and Haemophilus influenza are two species that will be fastidious. So you will see these names again later. Also, the conditions you incubate the plate in matter. So when we say we're incubating at 37 degrees in our room, that is aerobic conditions. They have access to oxygen. Some species are killed by too much oxygen. We have to be mindful of that as well. All right, so the materials you need for Wednesday, you need one ESNY methylene blue plate. So remember that E and then the MB are referring to two different dyes that are both the indicators and the selective agents. You also need one blood agar plate. So not lysed blood cells, these are whole blood cells. You need your inoculation loop, your incinerator. We're not going to use the vortex because I'm going to give you plated cultures. You're going to need your marker. And again, not broths, you're getting plate cultures. You also will need your unknowns that you streaked out yourself this time, so hopefully they're not contaminated. All right, so protocol for EMB, you're going to label your plates as usual. Here we have 4 sections. You need to divide your plate into 6. So I will do that for you on the board in class, but you will be making 6 sections because you also have to include your 2 unknowns. You're going to transfer an organism just as you did last time. If the plates look wet, depends on who made them, you're just going to do a little line in the center of each section. If not, you can zigzag like this. We'll see when we get there. And then for your blood agar, same thing. This one will have 5 sections. You have the 3 organisms here, and then your 2 unknowns. So you're going to label the plate. You can write blood agar on it. It should be pretty obvious. If you don't have room, don't worry. But make sure you put at least your initials on here and your section number so we know who to get the plate to in case it gets mixed up. And so that's it. That's all you need for your day 2 of selective media and your first differential plate. So remember, you've got EMB and blood agar, and that is all you're inoculating. So hopefully you'll be done fairly quickly. You can get out and spend your time studying.